Hello, everyone. Thanks for coming on a Sunday. It's my pleasure and honor to introduce Dean Abecassis, the Dean of the School of Medicine, who really, through his support, all of this is possible, support the Sarver Center, Cardiology, and research, and we're able to invite our colleagues from Stanford to enlighten us about their research in the next couple of days. Dean.
Thank you very much, Hesham, and thank you all for coming. This is the second annual Sarver Heart Center Symposium, and I'm really excited that, that you're all here to do this, and I think we have some really exciting presentations. He told me what to say, so I want to make sure I'm not forgetting something. Not really, but yeah. So this symposium was launched to try to foster collaboration between our cardiovascular center and other cardiovascular research institutes and people that are seeking to advance the field and the science. And so it's always great to have like-minded people get together and try to figure out what the next step is. So this year, we welcome colleagues from Stanford, from the Centro Nacional de Investigaciones Cardiovasculares in Madrid, the CNIC, and the College of Medicine in Phoenix, and of course, the College of Medicine Tucson. I think that you will see that they will be presenting some cutting-edge discoveries in basic science and translational science and AI-aided drug discovery, heart development, cardiovascular epidemiology, and atherosclerotic plaque imaging, to name a few. So very interesting topics. I'll finish by saying that we are extremely honored to welcome Dr. Joseph Wu, the director of the Cardiovascular Research Institute at Stanford, as our keynote speaker today. And from a personal perspective, I would like to thank Dr. Wu. I think it was about 2 or 3 years ago, actually at the Guder Foundation event, I sat at his table and I said, can you please help us get a director for the Sarver Heart Center and chief of cardiology? And he said he would. And so I thought, sure, you know, he's just saying that. But he was very instrumental in us being able to recruit Dr. Sadek. So at least from that, experience, and from what I've heard, he is a man of his word. So I want to thank you, Dr. Wu, for your support and for helping us. We wouldn't be here if it weren't for your help. So thank you. Did I miss anything? Thank you. Okay. Have a great symposium.
Thank you so much, Dean Abecassis, for the opening remarks. So without further ado, I'd like to introduce my friend and colleague, Dr. Joe Wu. So as you can see from the program at the tables, we can introduce Dr. Wu for the next hour, instead of him speaking, talking about his accomplishment and his accolades. As many of you know him very well. I'm just going to try to focus on something. So if we want to talk about awards and grants, everything from the NIH Transformative Award, the New Innovator Award, the Presidential Award, AHA Established Investigator Award, multiple invited lectures and honorary lectures. He really, I would say, he's the physician-scientist. As they say, doctors, or a scientist. He embodies the spirit of a physician-scientist. He is someone who is very active clinically very active in research. He has a very big lab that publishes quite often and a lot, and very high impact papers. His H-index score is close to 150 now, I think. He's a top 0.01 published investigators in the world. What's really important about Joe's work is it's not only mechanistic, and it's not only very deeply scientific, but also his work is always focused on the clinic, always translational, always looking for drug discovery. He developed and runs the biggest IPS platform, IPS cardiomyocyte and IPS platform in the world at Stanford, which he built. He is also credited with multiple discoveries, including the idea of clinical trial in a dish, whereby, for example, if you want to test a new drug, you don't have to go to large animals anymore. And the FDA adopted that based on his work, that now you can do a lot of these studies in cellular models, which he helped develop. I think also he has an eye in the clinic, not only in his research, but all through several companies that he participated in. And he has, I think, 2 or 3 drugs that are heading towards clinical trials or different phases of clinical trials. So he really is, I think, a role model for many of us, definitely for me. And it's been a pleasure to collaborate with him and to work with him throughout the years. And it's really a pleasure to welcome him here today. Thank you, Joe.
I want to set this up. So while I'm setting up, I just want to thank Dean Abecassis for inviting me here, and also let me just set this up. Hashem for inviting me here. I think for those of you who don't know, I got my real estate license when I was in high school. And in real estate, when you find a house, you get a finder's fee. But I'm still looking for the paycheck for the finder's fee for Hashem. But just joking aside, it's really on behalf of all our Stanford colleagues who are here. We're very honored to be here, to be able to come and participate in this second annual symposium. Hopefully there'll be many, many more to come. While we're going to get this set up, maybe you can help. Yeah. So I think Hashem and I go back a long way, and to me, it's really gratifying to see Hashem come and build up this Sarver Cardiovascular Institute, and also build up the cardiology program. And I think this is really putting the U of A on the map. And I've said this many times to my colleagues at Stanford, and I always say, you guys got to look out for U of A, because they're building one of the top programs in the country. And you could tell by this event that's being held here. And I'm sure in 2 years, 3 years, when you come back to this event here, you will see a much bigger presence. And in another 2 to 3 years, you'll see even a much bigger presence. So with that, I'm going to talk about what we've been doing at Stanford and also in my lab. And the title here is on stem cells, genomics, and AI for drug discovery. And this is my disclosure slide here. So the talk will be broken into 4 different topics. One is on what I think are the key platforms in biomedical research. Another one is on examples of hit-to-lead validations. And another one is on the new alternative methodologies that I think all the students in the audience should be aware of. And the last one is on the clinical trials in the dish. In terms of the first one right here, the 4 key platforms, in my own personal opinion, for drug discovery, one key platform you should be aware is the recent advances in next-generation sequencing. This is getting much faster, cheaper, with lower error rate. Just to give you a reference, back in 2000, it cost you about $3 billion in 13 years for the Human Genome Project to complete the first human reference genome. Today, you could get your DNA sequence for less than $500, and the turnaround time will be 3 weeks. So I've got my DNA sequence. I'm not sure how many of you have got your DNA sequence. This is really a powerful technique. And this slide shows you how fast the field is moving. For example, Nature published the UK Biobank sequencing data set involving 150,000 patients in 2022. And then 3 years later, they published another paper involving 490,000 patients. In my opinion, the second key technology is the different types of genome editing methods, starting with zinc finger nucleases, and then with TALEN, and then more recently with CRISPR-Cas9. This is discovered by Emmanuel Charpentier and Jennifer Doudna in 2011. So you can now use these genetic scissors, including more recently base editing and prime editing, to change the DNA of animals, plants, microorganisms, and also stem cells with extremely high precision. And then in December 2023, the FDA approved its first CRISPR-based gene therapy called Casgevy. This is for treatment of patients with sickle cell disease. I was actually on the FDA Cell Tissue and Gene Therapy Advisory Panel that reviewed the data that was presented to us by Vertex for the approval of Casgevy. And then the third key technology, of course, is the human iPSC platform discovered by Shinya Yamanaka around 2006. Because these iPS cells are genetically identical to the patients, you now have a test bed to validate your human genetic variants that you identify for your next generation sequencing, and then you can then cut in and cut out using your CRISPR editing. And then just as an example here, for cardiovascular diseases, over the past 20 years, we've been able to develop a very robust protocol to differentiate these iPS cells into cardiomyocytes, fibroblasts, smooth muscle cells, endothelial cells, and cardiac pericytes, what I call the big 5 cell types in the human heart. So over the past 10 years, and more recently in collaboration with Greenstone Biosciences, we've also made iPS cells for more than 2,500 patients with different medical history, ethnicity, and sex. This is the world's largest biobank of human iPS cell lines. These lines are linked to medical information. We perform multi-omic sequencing on them to help us understand how does the human genetic variants impact the drug response phenotypes. We work with the FDA on drug safety testing, and we also share these cells with the broader scientific community. Today, we sent out more than 4,000 vials to more than 500 investigators so far. And then this is our typical workflow for patient recruitment. We recruit from diverse population of patients in our clinic. We then generate their 2D micropattern human cardiomyocytes. We can also generate their 3D cardio-organoids and also 3D engineered heart tissues. Besides the cardiac, we could also generate the iPSC-derived liver cells, iPSC-derived skeletal muscle cells, and iPSC-derived islet cells for our drug discovery. And then show here some examples of iPS cell lines we generated from various types of cardiovascular diseases, as well as the associated genetic variants. For example, in the case of dilated cardiomyopathy, we have actin, lamin, myosin heavy chain 7, troponin T, and titin mutations. In the case of troponin T, about 15 years ago, we recruited a 3-generation family with troponin T mutation involving arginine to tryptophan switch. And then we're able to show that compared to the healthy control cardiomyocytes, the diseased cardiomyocytes had altered calcium ion, decreased contractility, and also abnormal distribution of the sarcomere alpha-actinin. And importantly, you could treat it with the beta blocker, for example, metoprolol, to improve the function of these DCM cardiomyocytes, recapitulating results from multiple large beta blocker clinical trials. This work was done by Ning Sun, who's now a faculty in China. For hypertrophic cardiomyopathy, our biobank has patients with myosin binding protein C, myosin heavy chain 7, ryanodine receptor, cardiac troponin I, tropomyosin, and so forth. In the case of myosin heavy chain 7, about 15 years ago, we were referred a large family with familial hypertrophic cardiomyopathy, as shown in this family tree. Through a whole series of experiments, we're able to show that this particular mutation, myosin heavy chain 7 with arginine to histidine switch, causes increased intracellular calcium, which leads to calcium handling defect, which then leads to cellular arrhythmia. It also causes activation of the calcineurin and fat signaling pathway, which leads to hypertrophic response. Importantly, when we treat it with calcium channel blockers, or beta blockers, as shown here, we could use it to reverse both the cellular arrhythmia and also the cellular hypertrophy. And this work was done by Dr. Feng Lian, who's now a faculty in China. And similarly, for left ventricular non-compaction, there are several genetic variants that can cause this syndrome, including calsequestrin, nebulin, phospholambin, T-box transcription factor, and troponin T. In the case of TBX20, about 10 years ago, we were referred this large family cohort at Stanford by our pediatric cardiology colleague, Dan Bernstein. Through a whole series of experiments, we're able to confirm that the mutations in the cardiac transcription factor, TBX20, can cause abnormal activation of the TGA beta signaling pathway, which recapitulates a key aspect of the LVNC pathophysiology. And this work was done by Kazuki Kodo, who's now a faculty in Japan. So there are many, many other examples of cardiac and non-cardiac diseases that we have. And as I mentioned, we'll be happy to share these IPS lines with all of you. And then finally, the fourth technology is really the recent advances of AI and computational platforms. As you may know, the 2024 Nobel Prize in Chemistry was awarded to David Baker at University of Washington for computational protein designs. The other half jointly to Demis Hassabis and John Jumper, both of them from Google DeepMind for protein structure prediction. And in parallel, the 2024 Nobel Prize in Physics, this was awarded to John Hopfield from Princeton University and Jeffrey Hinton from University of Toronto for foundational discoveries that enable machine learning with artificial neural networks. So this is a recent review article that I co-authored that discussed the various aspects of AI-based drug development. These steps can include target identification, drug discovery, preclinical studies, clinical studies, and also regulatory agency review, as well as post-marketing surveillance. One example of post-market surveillance would be, for example, using your Apple Watch to monitor your cardiac arrhythmias. The first two initial steps are most relevant to people in this audience here. I think most of us are basic scientists. And that is target identification and also discovery of active compounds, followed by lead optimization, which I will discuss further. Next, I want to show you some examples of hit-to-lead validations. As you know, the process of drug discovery is both time-consuming and expensive. For example, it typically costs you $1 to $2 billion and about 10 to 15 years to bring a new drug from the discovery phase through clinical trials to market approval. So this is a major focus of research in my lab. So in collaboration with computational scientists at Greenstone, my lab routinely uses AI machine learning for drug screening, also for molecular docking campaigns. For example, here we could use the FDA repurposed library, the Zinc22 library, the Wuxi library from China, the Enamine library from Ukraine, or the Greenstone virtual library, which consists of about 1 billion compounds. Alternatively, we could also use the Stanford library, which has about 100,000 physical compounds. Here, we can screen a diverse chemical library against both the target protein and also problematic off-target proteins using AutoDoc-Vina. The top 50% of the binders are then refined using Boltz-2 software. The toxicity properties are then predicted by using our AMIT AI software, available as free software on the website. And then the binding scores, as well as the AMIT properties, are then combined into a multi-dimensional analysis, and then the compounds are then ranked. The top 100 compounds are then evaluated using molecular dynamics simulation in GORMAX, and then the top ranked compounds are then further evaluated, as shown next. So this is a typical discovery workflow that goes from in silico to in chemical to in vivo target identification. For example, in step 1, we would use an iPSR reporter line with some type of reporter gene, in this case here, ACTA2 driving clover. The cells are cultured in 2D monolayer and they're differentiated in the cardiac fibroblasts. Here we're looking for anti-fibrotic drugs. We then screen for about 5,000 compounds at 7 dose concentrations. We give TGA beta, and then the TGA beta would cause the CLOVER2 color to shoot up. Here we're looking for compounds that can cause the CLOVER2 color to come down. The EC50, effective dose 50, and the TC50, the toxicity dose 50, can then be calculated by a dose response curve. Excuse me, at step 5, the initial candidates are then filtered to obtain compounds with satisfactory absorption, distribution, metabolism, excretion, and toxicity profiles. At step 6, the top 20 compounds are then subjected to counter screen in iPSC-derived cardiomyocyte endothelial cells. At step 7, the antifibrotic effects of your top hit can then be validated by a series of functional tests in multiple iPS cell lines and also in 3D engineered heart tissues. At step 8, the therapeutic effect of your top hit can then be examined in different mouse models of cardiac fibrosis and heart failure. And at step 9, the molecular target of the lead compound can then be identified by surface plasma resonance, by in silico docking, by SAR, by molecular dynamic simulation, and also by single-cell sequencing. Next, I want to give you some examples of how we incorporate stem cells, genomics, CRISPR, and AI for drug screening. As one example here, we use the iPS cells from breast cancer patients to recapitulate doxorubicin-induced cardiotoxicity, or DIC. Some of you may know doxorubicin can cause cardiotoxicity in about 10% of the breast cancer patients, but the problem for us as clinicians is that we don't really exactly know which 10% will develop cardiotoxicity. This work was actually motivated by my late mother-in-law, who developed breast cancer, received doxorubicin, and her LVEF dropped from 65% down to 45%. To study this, we recruited breast cancer patients. A year later, some of them had normal cardiac function, and some of them had doxorubicin-induced cardiomyopathy. We generated the iPSC cardiomyocytes, exposed them to doxorubicin in vitro, and here, as a proof of principle, we showed that if we pick the patients appropriately, their iPS cells can be used to recapitulate which patients are more susceptible to DIC. This work was done by Dr. Paul Burrich, who's now a faculty at Northwestern. And in a more recent follow-up study, we then used CRISPR interference and CRISPR activation to look at roughly about 2,300 genes that encode for kinases, phosphatases, and other druggable proteins that are involved in DIC. So using CRISPR-I, we found that if you inhibit the gene carbonic anhydrase 12, or CA12, you can rescue the cardiomyocytes from DIC. And using a molecular docking approach, we further identify a small molecule called inducilam that can inhibit CA12 activity and mitigate DIC. And as you can see here, this is a control mice with normal EF. After treatment, the EF goes down, and after treatment with the inducilam, we can rescue the LVEF. So this is an example using these powerful CRISPR techniques to screen druggable targets. This work was led by Dr. Chun Liu, who's now a faculty in Medical College of Wisconsin. A second example is shown here. I think, as you know, marijuana, this is being used increasingly in the US, legal in about 40 states for medicinal use, 24 states for recreational use. Interestingly, if you look at the UK Biobank database, you actually can definitively show that marijuana use is associated with increased myocardial infarction, but the mechanism is not well known. Here we show that the delta-9 THC, this is a psychedelic component of the marijuana. It binds to the cannabinoid 1 receptor in the brain to give you the high. It also binds to the cannabinoid 1 receptor in the peripheral vasculature to give you the vascular inflammation, the oxidative stress by activating MAP kinase, NF-kappa B, and cytokine pathways. So we now use AI in silico chemical library screen. We found that ginestin serves as a neutral CB1 antagonist that can competitively block the binding of THC to the CB1 receptor peripherally. Ginestin does not cross the blood-brain barrier because we don't want to mitigate the psychedelic effect of THC, and this is shown in this cartoon here. Interestingly, ginestin is actually a flavonoid found abundantly in soybean. And so my former postdoc, Mark Chandy, who was the first author on the paper, is now a faculty in Canada, is now initiating a clinical trial. He's already recruiting patients by giving chronic marijuana users with genestin to see if it can mitigate the vascular inflammation. Here's the third example, and this is led by Dr. Hao Zhang. He's actually leaving my lab at the end of this month to join UCLA as a faculty. As cardiac researchers, we know that cardiac fibrosis impairs cardiac function, but yet there are no good clinical therapies exist. And as shown in this cartoon here, mechanistically, we know that TGA-beta can stimulate fibrotic response by canonical SMAD and as well as non-canonical ERK pathway, which upregulate the extracellular matrix expression. However, these extracellular matrix proteins can also worsen fibrosis by stimulating the MD2-TLR4 pathway, which constitute a positive feedback loop through the TGA-beta. So using in silico and chemical simulation, we look for drugs that can block this interaction, and we found that artesunate came up as a hit, as shown in this docking screen here. Now, artesunate is actually a derivative of artemisinin and dihydroartemisinin. These are the primary drugs that are used to treat malaria, and it has saved millions of lives. In fact, the Chinese scientist who discovered this, Dr. Tu Yu, she received the 2015 Nobel Prize in medicine for this particular discovery. Now, because artesunate is an FDA-approved drug, we can then try to repurpose for anti-fibrotic indications. So recently, we also received approval from the FDA to test whether artesunate can be repurposed for treating patients with idiopathic pulmonary fibrosis. In a phase 1 study at Stanford, I will be the sponsor, and Dr. Josh Mooney will be primary investigator for the trial. This is a dose escalation study that we will perform. Here's the fourth example. This is led by my former postdoc, Shane Cho, who's now a faculty at Johns Hopkins. As mentioned in the last slide, in damaged heart, the extracellular matrix is actually a very strong mechanical sensing signal that can further promote fibroblast activation by a positive feedback loop. So to look for additional potential therapeutic targets, we first analyzed single-cell RNA sequencing data sets from both human and mouse hearts, as shown here. We identified a stroma-enriched mechanical sensory kinase called SARC to be a key player. We then performed a large-scale in silico docking screen to identify a top candidate of SARC inhibitor called serakitinib. And as shown in this cartoon on the left, when we treated the activated fibroblasts with the dual therapy that has serakitinib and TGA-beta inhibitor, we found that this combination therapy can actually shift from an activated myofibroblast state back toward a more quiescent fibroblast state, as summarized in this cartoon. Now, finally, this is the fifth example I'll show you. This involves a recent project on COVID vaccine. And I think, as you all know, the Pfizer-Moderna mRNA vaccine play a significant role in controlling the COVID pandemic. However, in a very small number of patients, they can develop myocarditis, mostly in young males after the second dose. But the mechanism is not well known. And here we analyze the proteomics data sets from patients who receive vaccines. Two cytokines that stood out, CXCL10 and interferon gamma. They were upregulated after vaccination in healthy volunteers as well as the patients with COVID myocarditis, and show much even higher in patients with the COVID myocarditis. We then showed these cytokines can activate inflammation in iPSC-derived macrophages and also can cause injury to iPSC-derived cardiac organoids. We then performed a loss-of-function studies by neutralizing these two cytokines in vaccinated mouse model, which then significantly reduced cardiac damage and reduced serum troponin serum cardiac troponin levels. We also showed that genestin, which I mentioned earlier in the marijuana story, can provide protection. And so, in summary, after the COVID vaccine, the T cells, as shown in this cartoon right here, the T cells can pump out interferon, and the myeloid cells can also pump out CXCL10. Together, these two cytokines can then cause inflammation in some of the individuals that we see in the clinic. This can then be mitigated by genestin in the mouse models. This work was done by Sue Kell, an instructor in my lab, and also Masa Nishika, who's now a faculty at The Ohio State University. Next, I want to talk about the recent developments in new alternative methodologies, or NAMs. Some of you may know the FDA Modernization Act 2.0. This was passed by both the Senate and the House in December 2022. The law authorizes the FDA to use alternative testing platforms such as stem cells, organoids, and in silico models. In April 2025, the FDA announced plans to phase out animal testing requirement for monoclonal antibodies and also other drugs. Specifically, the FDA's animal testing requirement will be reduced, refined, and potentially replaced using a range of approaches, including AI-based computational models of toxicity in cell lines and organoid toxicity in what's so-called a new approach methodologies, or NAMs. In April 2025, the NIH also made an announcement to prioritize human-based research technology by focusing, for example, on organoid, tissue chips, and other in vitro systems that allow scientists to model human diseases and capture the human variability. That's a key word right here. Capture the human variability in patient-specific characteristics. Another focus is on computational models to simulate complex biological human systems, disease pathways, and drug interactions. And in September 2025, the NIH announced a plan to set up a standardized organoid modeling center at the Frederick National Lab for Cancer Research, with contracts totaling $87 million for the first 3 years. And then finally, in November 2025, the UK also announced a roadmap to implement alternative methods and to reduce animal testing. For example, by 2027, researchers are expected to end tests of the strength of Botox in mice, and by 2023, it would also reduce pharmacokinetics studies on dogs as well as in non-human primates. So if you're interested in following the development of NAMs in recent years, here's a commentary that we wrote that discuss why journals, funders, and also researchers should understand what NAMs is all about and think about this in your day-to-day scientific research. This slide highlights, in my opinion, what I think is one of the major problems for the high failure rate in drug discovery in basic science research, and that is the lack of genetic diversity and pharmacogenomic differences with traditional models, such as using immortalized cell line and using inbred mouse. For example, because they're immortalized, this is essentially the same as n equals 1 right here. And the deviations that you see here is not the biological variability, it's actually the pipetting skill of a graduate student and the tail vein injection skill of a postdoc right here. What I really want to know before I go to a clinic is what is the biological genetic variability that exists among all of us. This pharmacogenomic difference is, something that you need to understand before you go into clinic, and this is otherwise impossible to recapitulate using a traditional immortalized cell line inbred mouse model. So shown here are 2 examples highlighting the inter-individual heterogeneity of drug response. The first example involves 4 different patients treated with 4 different kinds of statins: atorvastatin, lovastatin, simvastatin, fluvastatin. Statins are cholesterol-lowering medication. I take it myself. I take atorvastatin. But they also have pleiotropic effects. And here, as you can see here, the RNA sequencing of how each one of the 4 patient iPS cell cardiomyocytes respond to statins is very different. Another example involves inter-individual heterogeneity to nicotine in iPS cell-derived endothelial cells involving 20 patients, 8 female, 12 male. Here, you can see that nicotine, in general, causes a shift to the left on PC1 and upward on PC2, right? But what's interesting is that you'll see that some of these patients responded quite well, whereas others don't respond. So let's say if you take blue right here, if you're Pfizer and you're using this blue cell line, you will see great response and you will go all in. On the other hand, if you're Merck and you're using this red cell line and you saw nothing, then you would say this is not a good drug for us, right? The truth is somewhere in between, as shown in this plot right here. Now, to drive home this point, this is the third example involving another 20 patients, iPSC-derived cardiomyocytes exposed to tyrosine kinase inhibitor called sunitinib. The figure on the left shows the differential gene responses on PC1, PC2 again. Now, if we zoom in to individual patient, here you can see that this patient, 955, the AHNAK, the EMC10, the LRRTME3, the PTPRK, the SNYE2, and the Titin are upregulated after sunitinib treatment. But which one's a target? Assuming sunitinib is the drug that you want to go with, assuming this is a completely new drug, you want to figure out what the target. Which one's the target? Well, there are potentially 1, 2, 3, 4, 5, 6 targets. What about if you have 2 lines, 955 and 2736? It seems like we have 2 additional targets pop up, ATP2A and CACNB2. What about if you have 3 lines, 955, 2736, and 2240? It's almost like going to optometrist. It's showing, can you see this? Can you see that? What about 4 lines? Well, 2159 doesn't really respond much to this particular drug. What about 6 lines? 7 lines? 8 lines? 9 lines? What about if you have 20 lines? Here, based on the data from 20 patients, we would most likely go after EMC10 as a target, right? So I hope this gives you an idea why it's important to have many, many different cell lines that you could use as surrogates for patient heterogeneity in your preclinical drug discovery. Now, the heterogeneity in the iPSC data set is not surprising because we also see the same trend in real-world clinical data. Many of the FDA-approved drugs in the market today actually do not work so well as you think. Shown here, for example, are the top 10 highest sales of drugs in the US. Blue means the drug works, and red means the drug does not work. So for Abilify, which is used to treat schizophrenia, out of 5 people, the drug only helps 1 person. For Nexium, which is used to treat heartburn, out of 25 people, the drug only helps 1 person. For clinicians, we call this NNT, number needed to treat. And typically, actually, most cardiology drugs, the NNT will be around 20 to 30 to even 40 for beta blockers, ACE inhibitors, and so forth. And so for the trainings in the audience, I think some of the challenges and opportunities for human-based research involving NAMs would involve, for example, understanding the throughput versus the tissue maturation, the vascularization of the perfusion, the scalability for industrial and clinical applications, the host transplant function integration, right? These are all perfect areas for the trainees in the audience to work on. For the trainees in the audience, other questions may include the genetic background, the cell line variability. I already mentioned this. In basic science, academia research, we don't want variability because we want a paper to get published ASAP. In drug discovery, in which you're thinking about clinic, you want to understand the variability, completely different MO right here. You want to understand the extracellular matrix and composition mechanics. I talk about this with the Stertzer story. You want to figure out a way to optimize universal multi-cell type media. Nature has a universal media, which is our body's serum and plasma. But in stem cell research, we still don't have a universal media. You want to do 3D imaging of real-time functional assays. And finally, integration with different organs on a chip system. And so recently, to address the question of multi-organ interaction, we were able to publish on generation of vascularized cardiac and vascularized liver organoids. This is a culmination of about 5 years of work. Here we screen about 35 different combinations of cocktails that are vascular-inducing factors to give rise to vascularized cardiac and vascularized liver that are spatially organized. These vascularized cardiac organoids have about 15 different cell types that are present in the human heart, including macrophages. And then the vascularized liver is important because, as you know, when you take a drug orally, the drug is the first pass metabolism is typically the liver, and the liver then basically convert the drug to metabolites. So here we want to put the drug in the liver organoid, take the metabolites and put it in other organs to simulate what's going on in vivo. And then for the last part of my talk, I want to just conclude with examples of what we call clinical trials in a dish. You should be able to do clinical trials in a dish in the future. So here I want to touch upon how you should be able to take patients in this room, generate your iPSL, develop your cardiac organoids, and then screen for drugs to see what drug works and what drug does not work before giving it back to you in real, clinical studies. I think this concept is ideal for developing new drugs or repurposing existing drugs for rare orphan diseases. And as some of you may not know that there are actually about 7,000 rare orphan diseases, and yet there are only about 770 orphan drugs that have been approved by the FDA. The idea is that instead of using the patients as a guinea pig, we can then generate the iPSL from the rare orphan disease patients, test which drugs work and which drugs do not work, and then initiate a phase 1 clinical trial that way. This slide is given by my colleagues at the FDA. So for rare orphan diseases, the FDA recognizes the challenges of developing new drugs due to small number of patients, and yet there are thousands and thousands of genetic variants for each disease. So now their approach is to accept data generated from cell models to test drug efficacy instead. As one example, cystic fibrosis is drug Callidico. This is made by Vertex Pharmaceutical. Initially approved for 10 genetic variants, and now has been expanded to 24 more genetic variants. Another example is Fabry cell disease. This is a lysosomal storage disease passed on by the X chromosome. The drug Galafold, made by Amicus Therapeutics, initially approved for 40 genetic variants, and now has been expanded to 348 genetic variants based on cell models. So currently, the FDA is interested in expanding this cell-based platform across the full spectrum of diseases. This slide is also given to me by the FDA. I think the value proposition here is using the in vitro efficacy data to reduce the number of clinical studies, to inform the regulatory decision-making at the time of marketing application, to streamline the drug development process, and to expand the indications based on in vitro data. Now, at Greenstone, we have also generated the world's largest biobank of rare orphan diseases. Greenstone currently has about 550 different rare orphan diseases. And if you're interested, you're welcome to reach out to them. They'll be happy to send these iPSL lines to you. So I want to finish up by going to this case report of a patient that we saw at Stanford, actually referred to us by my colleague, Paul Wang, 57-year-old man, previously healthy, came to the clinic, found to have severely reduced left ventricular ejection fraction. Family history is remarkable for multiple sudden cardiac death, heart failure, hypertension, prompting a clinical genetic screening. His genetic panel shows a pathogenic variant lamin AC. Question, what is the mechanism of the disease? Question, how does the genetic information affect his management? Question, how do we model the disease using the human relevant assays I just mentioned? And how do we treat it outside of standard care, clinical trial in a dish? So it turns out the family belongs to a large 4-generation family cohort with the lamin AC frameshift mutation that leads to early termination. As you can see here, many of the affected individuals, they had cardiomyopathy, atrial fibrillation, atrial ventricular block, and importantly, 8 of them dropped dead. 8 of them dropped dead from sudden cardiac death. And so this is a horrible toll on the family. And so here, Jay Cho Lee, who's now a faculty in Korea, figured out that the mechanism is due to abnormal activation of the PDGF in their cardiomyocytes, which leads to abnormal cellular arrhythmia and also impaired contractility as shown in this cartoon. We also showed that if you block the PDGF pathway with the PDGF inhibitor called quinolinib at low dose, you can rescue the abnormal phenotype of these mutant cardiomyocytes. However, quinolinib is actually a Pfizer drug. They licensed it to AROG. AROG is using it for ALL, AML. They're in phase 3 clinical trial. And they weren't interested in sharing with us because whatever data that we have can impact the FDA submission. So we weren't able to use quinolinib and repurpose it for our patients. Now, but what was interesting to us is that many of these patients at a young age actually develop high blood pressure. And usually if you have a heart failure, your blood pressure is on the low side, not on the high side. And so we were quite puzzled by this finding, and we decided then to generate the iPS cell-derived vascular endothelial cells on the family cohort. We compare the disease versus the healthy. To make a long story short, we identified KRF2 as a transcription factor that is responsible for the endothelial dysfunction. And then this time we used the FDA-approved drug library screen. We found that lovastatin can be used to activate KRF2 and then can be used to rescue the endothelial dysfunction. In contrast, lovastatin is FDA approved. We can now just give it to our patient. That's exactly what we did. We actually give it back to the patient, not to treat the cholesterol, but to treat the vasomotor tone. Essentially, we use a machine called EndoPad. You occlude the brachial artery for 5 minutes, you release it. Once you release it, for healthy individuals, the peak flow over the basal flow should be 1.8 and above. These patients, surprisingly, when you release it, the peak flow is actually lower than the basal flow at about 0.8. And then after treatment with lovastatin for about 6 months, we're able to show the ratio becomes about 1.2, 1.3. Not normal, but significantly improved compared to 0.8. And this is also the reason why the title here is Clinical Trial in a Dish using iPS cells. So I believe the FDA Modernization Act 3.0, this will really pave the way for the next area of drug discovery. It doesn't matter whether you're using predictive AI or generative AI to find the optimal candidate, similar to the stories I showed you earlier with Genestin as a neutral CB1 antagonist, altusinate to block the AD2-TLR4 interaction, serakitinib as a mechanical sensory kinase inhibitor. As shown here, these drugs, or peptides, or antisense oligonucleotides, or RNA therapeutics, and so forth, are then tested in your traditional in vitro assays, followed by in vivo animal studies. Afterwards, a clinical trial in a dish model can be used to help you predict the safety and efficacy of your lead compound prior to testing them in patients. So let's say, for example, you have drug number 1. Drug 1 fails. Drug 2 fails. By the time you go to drug 3, the investor already pulled out. On the other hand, here at a fraction of a test, I can tell you that drug 1 works in 1 out of 1,000. You're not going to pursue that. Drug number 2 works in 1,000 out of 1,000 patients. That means my scientists made up the data. There's no way a drug is that good. Drug number 3, it works in 300 out of 1,000. Yeah, that's something that's reasonable. That's something that we will want to go with. And that's a drug that we choose to go to clinical trial. So in summary, I hope I've shown you the framework of how there may be a paradigm shift in the future using these iPS cells to help us understand first the molecular mechanisms of diseases, the biological specimen, or what I call the biological data, can then be converted into multi-omics digital data using multi-omics sequencing, pathway analysis, and drug target profiling. The digital data can then be fed into AI machine learning software for molecular dockings, molecular dynamic simulation, in silico amide prediction. This will then give us drugs that are much more precise and predictive. The lead compound can then be tested on the patient-specific iPS cells that have the desired disease phenotypes, not immortalized cell lines, but the iPS cell with the disease that you're going after, hence completing the loop of clinical trials in a dish. The ultimate goal is to decrease the cost of drug development, shorten the timeline, and develop therapeutics that are more efficacious and safe for our patients, the holy grail of precision medicine. Now, if you're interested in what I just talked about, Stanford actually holds an annual drug discovery symposium. We've been having this for the past 10 years. This is the symposium from last year. And then this year's symposium will be April 27, 28. And within these 2 days, we basically bring the CEO, CSO, and CMO of Big Pharma. We bring in the VC and also the FDA, the NIH. And over 2 days, we talk about how do we accelerate drug discovery. And lastly, if you're interested in any one of these cell lines, you're welcome to reach out to Greenstone Biosciences. They can give you these cells free of cost, and you just need to email them, and then they'll be happy to provide you. As I mentioned, we've given out more than 4,000 vials to more than 500 investigators so far. And so this concludes my talk. I want to acknowledge my postdocs, my graduate students, instructors, my collaborators, my former trainees whose work I highlighted here, and also funding support from the NIH, AHA, Chen Zuckerberg, FDA, and the Goodell Foundation who help us support some of our missions. And then I just want to conclude by saying that I've been very, very, very impressed by UFA over the past few years. I think some of you may know my predecessor at the Stanford Cardiovascular Institute, Dr. Bobby Robbins, was the president. When he left at Stanford, I took over his position. So I've been coming to UFA for many years. And there's a bond, there's affinity, I guess there's a love for UFA on my part. And I'm really glad to see that the leadership under the Dean, under James Liao, under Hashem, under Hossein, and many other people, Hasakti, Joe Tadif, and many other people are building this place into really what I would consider one of the top places in cardiovascular research. So thank you, and kudos to you, Hashem. Yeah, thank you very much. Yeah.
Wow. Thanks so much, Joe. This is really amazing. Every time I hear you talk, I see more nature and science in Cell papers, and it's kind of depressing. But congratulations on fantastic work. So I'll start with asking a couple questions. How do you reconcile the fact that some of the phenotypes we look for cannot be recapitulated in a dish? So for example, effect of hemodynamics, effect of stretch, mechanical load, arterial stiffness, all of that. How do you reconcile that, and how do you integrate that into your screen?
Yeah, so that's a very good question, right? So the cells that we're generating, either 2D or 3D, they're not the same as humans. And some of my colleagues, I actually disagree with my colleagues, right? Some of my colleagues, they say that you could completely wipe out animal testing, which I don't think is the case. I think you can reduce the number, but you cannot wipe them out. With regard to the contractility, with regard to shear stress, shear flow, and different kind of functional readout, which is more difficult, I think the transcriptomic, the multi-omic data is actually better than the mouse data. But the functional readout, let's say, for example, the power output at the heart is this much, and within the organelle, you're generating this much. It's not the same, but we're not interested in matching it exactly. What we're interested in is understanding baseline with stress and with drug. That's what we're interested in. The scale that we're working with is much on the lower end compared to the actual human heart that's working on this level right here. So you could model this with shear stress as long as you have a basal number, and as long as that basal number goes up or goes down with some kind of stress, and as long as you have a drug that's positive control that can show it mitigates or improves, then you're ready to start doing your drug screen. Once you do all your drug screen, then the drugs will still need to be tested in, in vivo animal models. The reason why I say that it's never going to replace is, as you pointed out, the 3D complexity, right? So I'll give you an example. So we're developing a drug for chronic pancreatitis, and the drug works beautifully well in mouse models, and we spend a lot of money showing that it works in mouse. You give it orally. But then the FDA wants a large animal model, so when we give it to a dog, the dog actually vomits. And then because the dog vomits, we cannot detect the blood level in the dog. And so we realized that the drug is irritating the stomach lining. And so what we did was we did enteric coating of the drug to bypass the stomach, and then it gets absorbed in the duodenum. And once we started doing that, we could start seeing a significant spike in the Cmax and the PK/PD. That kind of stuff you can't model it right now with these iPSL organoids. Yeah.
Great. So the second question is related to orphan diseases. So why does the FDA still insist on human trials at all, right? So you can argue, for example, let's say Fabry's disease, for example, why don't they accept an assay like decrease in GB3 levels when you have these patients that don't really have much other choices? Maybe Fabry is not a good example. There's some, their placement therapy, but many of these rare diseases, there's absolutely no option. And, for something like progeria, for example, they forced the trialists to have a randomized control trial when there's only a handful of these patients around.
Yeah, so the whole mindset is now changing. I'm actually on the FDA's, not the FDA, the NIH SOM Center, the Standardized Organoid Center. But the whole mindset is completely changing now. So let's say, for example, if you have a rare orphan disease patient and you have a drug, and the drug is a repurposed drug, you don't need to go through all that stuff. You just need to test it on your organoid and to show that it has some kind of meaningful readout. You should be able to just go to the next step. There's a paper I can share with the audience right here by the FDA scientists. They've actually listed a significant spike in terms of the number of submissions to the IND package that are based on in vitro, in chemical, and silico models. Significant spike. So right now, depending if it's a completely new drug, they've never seen it before, then they would ask you to say, okay, can you show me what is the absorption, what is the CK, what is the PK/PD, what is the AMIT? And you will still need to do some of that. But in the future, you should be able to do less. Let's say, for example, all of us are PIs in this room. There are 200 drugs, and each one of us is saying my drug's the best, right? And to take each one of our 200 drugs, because 200 PIs in this room, hypothetically, all the students are considered PIs now for this argument. So if you take these 200 drugs and you put it on to do the validation, you'll probably need about 200 mice, right, to kind of get a sense of whether it works or not. So 200 times 200, my math is shaky, but I think that's about 4,000 or something like that. Oh, actually 40,000. 40,000 mice. Right? What we're saying is at a fraction of the cost, give us these 200 drugs. We could test it on a panel of these patients. Actually not on immortalized cell line, but on iPS cells from all of you on a dish. And I can show you that instead of 200, you should narrow it down to about 5. Take these 5, go to the animal testing. Now, all of a sudden, you're taking 5 times 200. Instead of 40,000 mice that you're killing, you're down to 1,000 mice. Yeah.
Then a final question for orphan disease. Do you even think if you're using a repurposed drug, do you need to go to a clinical trial, or you can just test it on patients? Let's say, for example, you have something that works in a dish, and it's an FDA-approved drug. Would you go the route of clinical trials, or would you just give it to
there are some N=1 clinical trials out there, and some compassionate use, and I would encourage you to talk to the FDA. My experience with FDA, they're actually very, very nice. They want to talk to you. They want to help you. And so if you have something that you think really works, set up a pre-IND call with FDA, explain to them, they'll give you some guidance, and based on the guidance, you can then quickly work on what you're trying to do. Yeah, so don't think of FDA as roadblocking. They're actually very professional, very collaborative people that you can work with, yeah.
Questions?
Hi, Joseph.
Yes.
Yes, impressive.
Antonio.
This is Tony Enriquez from Madrid. Yes. I really just want you to give us some thought on some questions of the lines that you are going, particularly in working in metabolic diseases. And metabolic diseases has a big problem that they really rely on the intertalk of not only cells in the same place, but also organs. And there is a second level of problem, that there are some genes that are expressed only in adult. For example, cardiomyocytes, we cannot bring them to very adulthood, and they don't express terminal genes that might be the cause of the disease. And this is one thing and the other thing is, if we work on mitochondria that use oxygen, the oxygen problem in all the IPS analysis is not under control. And it's not easy, because even if you reduce the oxygen to the chamber, maybe you reduce too much because the cell doesn't have the proper delivery, they don't have the hemoglobin, and they don't have all the systems to integrate. Are you thought on how we should really pursue it to improve the quality of the culture?
Yeah, very good question. We thought about it, and I don't have a lab that's so huge that we can tackle all the questions, right? So let's say, for example, I'll give you a reference. 20 years ago when I started my lab, we were one of the first to work with human ESL, human iPSLs at Stanford. We use a hanging drop assay and to get the beating cardiomyocyte. Within that beating embryo body, we're only getting about 1 to 2% cardiomyocyte 20 years ago. But because 1 to 2% cardiomyocyte causes the whole embryo body to beat, we see it, we take a video, we're thumping our chest and say, look, we're getting cardiomyocytes. 20 years later, we have high school students who come by, 8 weeks, they're getting their own cardiomyocytes, right? So the cardiomyocyte differentiation aspect, the hepatic, the liver, all these differentiation aspects, it's improved dramatically. The maturation aspect, which is what you're mentioning, let's say, for example, this is a human adult heart, and this is the iPSL cardiomyocyte. We're pushing it right here. I'm waiting for one of the smart postdocs or a graduate student in the lab to push it to here. It's never going to be here. It'll never be here because the in vitro culturing condition is never going to match the in vivo. Another way to say it is, if you take a liver biopsy from me today, you put on this dish, day one, it looks the same. By 7 days, you would have de-differentiated because the liver plastic conditioning, the cell culture is never going to match it. So right now, we're probably right here, and hopefully we can get somewhere right here, but we'll never be here. With regard to the oxygen issue, yeah, I mean, right now we culture, you can actually get healthier iPSL, you get healthier iPSL cardiomyocytes using hypoxia culturing method. Problem with us is that to have a hypoxia culture method, we got to run a nitrogen tank through it. And that becomes very, very expensive. And that's the reason why we don't do it. But if the cost is not an issue, I agree with you. Using a hypoxia model would be a better way to generate these cells and differentiate these cells. Yeah.
If I'm allowed another question.
Yes, yeah.
Yeah, the variability in the population that you are facing, that is very interesting. Did you ever thought in anticipate, not just wait for the diseased people, actually to make a screening in silico? With the variants that we know that are in the population, we are accumulating more and more information to really have any information about the stability of the protein structurally or the interaction with the drugs that might be secondary, and feed the AI with this information already to not to wait for the disease cases only, just to see that when you give the information of the genome of the diseased patient, they can also know, okay, guys, if you give that drug, you are actually have a secondary potential effect here.
Yeah. That's a very good question. Keep in mind that, in my opinion, I think different clinicians will give you different opinions. In my opinion, I think genetics account for about 10%. The 90% is our social, environmental, behavioral thing, right? You can see it clearly within the same family, two siblings. Genetically, they're 50% the same, but yet they're very different depending on what their personal preference are. The iPSL, in my opinion, is a great model to look at that 10% of genetics. You cannot predict what's going on with the behavioral issues. So we do have some projects in which we are exposing the cells to common cardiovascular drugs, aspirin, beta blocker, statin, ACE inhibitor, spironolactone, SGLT2 inhibitor, GLP drugs, and then single combination to see what is the effect of those drugs on endothelial cells and cardiomyocytes. Ideally, if I have unlimited resource, yeah, I would create a roadmap, an atlas, in which the atlas has 1,000 patients. And it's got, within the 1,000 patients, it's got 7 or 8 different major organs. And it's got the baseline, and it's got the drug response to 50 most common drugs that we're going to take as patients, right? There are common drugs that all of us are going to take. I bet you, all of you, not all of you, maybe 90% of you will be taking a statin, right? And so we could create that roadmap and just put it out there. We just don't have the budget to do that. Yeah.
One question, Joe. Thank you so much again for coming and giving this keynote lecture. And also we want to thank you and Greenstone for sharing some of the cell lines that we have in our lab. The question I have is, differentiation, maturation, and then performing functional or phenotypic characterization. I think there are several steps in the process, defining a phenotype in iPSCs. We do a lot of challenges like reproducibilities and consistency. Postdocs and trainees are really struggling. My question is, how do we standardize? What is the standard protocol, or like what is the reference we can go forward and have a systematic procedure?
Yeah, so that's a very good question. That's part of the reason why the NIH set up the SOM Center, SOM, the Standardized Organoid Center. Problem is they actually don't have experts. They asked me to see, hey, Joe, do you have postdocs in the lab who want to join to lead the cardiovascular, lead the liver, and so forth? They're going to set it up. And the way they set it up is they'll basically say this is a protocol that the NIH is using, and hopefully people will follow. I think the heterogeneity, the variability, right? People think about variability as a dirty word, right? Because as scientists, we don't want variability. The variability could be due to technical proficiency of your postdocs and graduate students and technicians. That could be trained. In our experience, most of the variability, because in our lab we have SOP, right? It's a standard operating procedure. In our experience, most of the variability is due to the genetic variability. And that's why I particularly show you what happens when 20 patients get treated with nicotine, 20 patients get treated with sunitinib, right? And so that's where most of the variability comes in. And somebody asked me this question last week when I gave a talk and say, how many cell lines should I use? And I kind of stumble a little bit, and I recover. I laugh and say, well, what is your indication? Are you using it for publication? The publication, you're a graduate student, you want to get out ASAP, and you just want 2 cell lines, 3 cell lines, and get a big p-value, and you want to get out and say that this works, right? But that's for publication. If you want to go for drug discovery, and you're willing to put in $50, $100, $200 million and say, I want to bet on this drug, you can't be doing 2, 3 cell lines, right? You got to use 20, 30, 40 to give you much higher confidence that my drug is going to work when I give it to everybody. Yeah.
Thank you.
Joe?
Yes.
Great talk. Thank you so much.
Oh, you want to go? Go ahead, and then I'll follow you.
Right here. It's a great talk.
Oh,
Thank you so much. I just wanted to follow up on the fact that when you take cells from patients and you turn them into iPS cells and then you turn them into cardiomyocytes or any cell types, these cells go through a lot of changes. And I'm surprised that doxorubicin-induced cardiotoxicity can actually be mimicked in iPS cells, because a lot of people think that what happens with doxorubicin is damage to the mitochondria. There's no genetic predisposition. Yeah. So how do you think this actually happens in a disease like doxorubicin-induced cardiotoxicity, that you can actually mimic it in iPS cells?
I think there are a lot of papers out there that have shown the genetic susceptibility, the genetic substrates, because I think some of the papers, I don't remember the exact name, some of these are different variants in terms of transport of the doxorubicin drug, influx and efflux. There's a whole bunch of papers on that. But you're right that when you make the iPS cell, you wipe out the epigenetics. And then also for that study, if you look at it carefully, I'm a clinician, so we realized that you cannot recruit patients who are history of heart attack, diabetes, hypertension, a whole bunch of comorbidities. So you don't know if the doxorubicin is due to the comorbidity or due to intrinsic genetic difference. So if you go back to that study, you'll see that most of the patients that we recruited are very young individuals with 0 comorbidities, no history of heart attack, no history of, hypertension, diabetes, no history of surgical, blah, blah, blah. And then we're able to show that you could see a difference. And I think many other labs have also, had similar findings that you could, if you pick the patients correctly, you could see a genetic difference of how they respond to a drug. Yeah.
Joe?
Yes.
Marv? First of all, I want to thank you again on behalf of everyone here for a tour de force. That was amazing. I have a question related to sort of policy and education, a little different direction than the other questions. So from your sort of perch of authority, how do you see this going? Because this truly is a paradigm shift. And we even saw the reaction of several of the prior questioners in the sense that each individual center can't do this. This is such a complex process that you really have to have centers of excellence that are going to be able to do this. And then you get into the split between academia versus industry. That's one issue. And then the mindset change for education. For our trainees, we have many people sitting here who are the next generation, and they're looking at this, amazed, but also glass-eyed in the sense that they can't individually do this easily in a lab. So on the sort of policy side, should we have centers of excellence? How do we incorporate this in education? Where do you see this going?
Yeah, so I think that's part of the reason why the NIH set up the SOM Center, the SOM Center. I think the initial investment by the NIH was about $90 million, and they'll put in additional money. But I just want to, emphasize to the trainees. I know there are a lot of trainees here. And 100 years ago, for the trainees, when we treat syphilis, venereal disease, we get the mercury. 100 years ago, right? And today is 2026. The question is in 2021, 26, people 100 years from now, will they be clapping at how great of a science that we're doing or how great a medicine we'll be doing? Or similar to the syphilis story, they'll be laughing at the way we treat cancer patients. They'll be laughing at the way we treat all the disease patients. They'll be laughing at the way we do our clinical trials, right? In the clinical trial, you open up any New England Journal of Medicine. I hope this is not recorded, but I don't say it in the public. You open up any New England Journal of Medicine, and you see two curves, survival curve and years and survival, and the two curves just overlap each other. P-value is 0.08. They say this is a bad drug. The same paper, the same journal, you see two curves overlap, but at the very end, it separates a little bit, and it becomes P less than 0.05. Then people are clapping, everybody should be on this drug. Right? In reality, is it really true everybody should be on this drug? It's not precise. I already showed you the number needed to treat. In cardiology, beta blocker, statin, ACE inhibitor, the numbers are 20, 30, 40. In fact, you have to give credit to the surgeons, right? The best one is the DVT prophylaxis. Number needed to treat is actually 8, right? And so I don't think the way we practice medicine 100 years from now will be the same as how we practice. I don't think the way we do research 100 years from now will be the same. The same reason, the HeLa cells, Henrietta Lacks cells, right? Somebody came up with it and just said we should use it. We should use it for cancer research. Somebody 50, 60 years ago came up with transgenic mouse and said, okay, we should use it. What I'm saying is that there's another tool, and these tools, I just showed you the 4 tools, are genetics, CRISPR editing, because you want to validate your genetics, iPSL, because that becomes a platform for you to validate it. It gives you a readout. Without the human iPSL, you don't have the readout. And then the AI. Right now, a lot of these AI could help you figure out what the drug design is. My pharmacology, my PhD was in pharmacology. Back then, we used to do a lot of pipetting, right? A lot of pipetting. But now the computer is smarter than us and look at this is a pocket, this is hydrophobic, this is hydrophilic, and there's a methyl group here, and the computer can kind of guess what kind of drug structure will fit that well, and the computer spits out all these drugs. Now, when the computer spits it out, you should not take it for granted, right? There's still a lot of false hits. You just need to validate it, but it's helping you to do that. So I do think that for the young trainees, I hope this lecture will kind of like open up your mind and say, hey, there's other techniques that we could adopt as the science moves forward. Yeah.
Thanks, Joe. First, I wanted to, particularly for the trainees, say how inspiring all of your work is and your presentation. And I think we think about science and the future of science. I think we can't but all be super excited about all of the promise you've talked about. You showed us, the comparable graph to Moore's law with whole genome sequencing, the cost being reduced. And then when you take your work and fast forward, 20, 30 years from now, precision medicine would be that everybody would get, after you're born, we would all get a whole genome sequence, and then we would probably all get differentiated by multiple cell types, including cardiomyocytes. What are the barriers to do that? Do you see that happening in 20, 30 years, that everybody, their cells will be differentiated? What are the big barriers that allow that prevent that today from being the high throughput kind of thing you would need to do that? But do you see that happening in the future? But thanks for your inspiring work.
I think it depends. If I have a crystal ball, I think it depends. I think in the US, our main limitation is reimbursement. Right? Who's going to pay for it? And it's possible 20 years from now, 30 years from now, everybody will get the DNA sequence, and everybody would have their blood banked, the iPSC generated. But in the US, the way we do our reimbursement, our healthcare delivery, I think it will be tougher. If I were to bet, places like Singapore, yeah, they may be able to do that, because there's much more government investment. If you have government investment, government is all vested and say that we want our citizens to have the DNA sequence, we want our citizens to have blah, blah, blah. That probably is the route to go. And you might become a scenario in which 20, 30 years from now, we in the US are actually looking forward to Far East and say, what are these guys doing? How can we copy them? Because it's just, in my opinion, they're just sometimes much more progressive than we are. Yeah.
Thank you so much for your interesting talk. So just a quick question. Since the vasculature is important in iPSC and organoid formation, did your lab or any other labs try to combine protocol for iPSC drive, for example, cardiomyocytes and blood vessels are iPSC protocol, since the vasculature and the flow is really important in these organs?
Yeah, so the protocol we published has a vascularized cardio organoids that you can follow. We're happy to share the protocol with you, but I hope 5 years from now, nobody's using that protocol. Because 5 years from now, 10 years from now, if people are still using that protocol, that means science has not advanced, right? So that's a starting roadmap. You could take that protocol and say, oh, this is what I add. It took us 5 years to come up with these 35 cocktails that we screen iteratively to come up with a cocktail that gives us cardiac and liver. You should take that and then add something to it to make it even better for your own application. But that one gives you the vascularized cardio organoids, yeah.
Thank you very much.
Any other questions?
So I've seen multiple methods for, you talked about maturation, how like we're here with iPSCs and how human like actual cardiomyocytes are over here. I've seen multiple methods to mature iPSCs, but a lot of times they come at the cost of throughput. Do you think do you think in the future it will always have to come at the cost of throughput, or there will be a way to just bring the iPSCs closer to our like cardiomyocytes without that cost?
Yeah, so the throughput issue will also be addressed. For example, we have a colleague at Stanford, Mark Schuyler Scott. He's got a big RBART grant, and he's generating these bioreactor of these cardiomyocytes at a very low cost, and he can generate billions. So the throughput is not an issue. The maturation is still an issue. And I'm looking at you, looking at other people sitting next to you to figure out, well, what is it that you need to add? Right? The way we're doing it is very brute force. We look at a frog, a chick development, and say, oh, okay, the chick development, this thing gets activated. I think let's try it right here, right? And so throughput, I think we solved that. The maturation is still something that we need to push, because once you're able to get mature, very mature cardiomyocyte, you can even use that to study aging, right? A lot of people are interested in the aging process. Right now, it's much more difficult to study aging for iPSC-derived cell type because the cells are immature. You could use it to study inflammation, right, by adding cytokine, but to study, or you could use it to study senescence because cells poop out, but it's much harder to study aging. So I'm looking at people here to help figure this out, yeah.
All right, so let's conclude the session by giving a big round of applause to Dr. Joe Wu. As you know, he's a clinician, scientist, administrator, and also leader in the field. I have no idea how much time he has to sleep. Maybe he works 24/7. It's such an honor to have you here with us today, and also as a keynote speaker, Dr. Wu. Thank you so much. We have a small token of appreciation. This is a very tradition in India. We do that. Oh, wow.
Thank you so much. Yeah, thank you. Thank you. Thank you. Thank you. Thank you so much. Yes, thank you.
So, if you want to take